Ready? Let's begin. The life sequence. In the temple precincts of ancient Sumer, stone carvers working around 4,500 years before the Common Era etched a symbol into sacred pillars that no one questioned for 5 millennia. 2 serpents, intertwined around a central staff, rising in a spiral. You know this symbol. You have seen it on hospitals, on ambulances, on the letterhead of every medical institution on the planet. What you were never asked to explain is why a Mesopotamian civilization with no microscopes and no molecular biology chose as their primary symbol of divine life, a structure that is visually identical to the double helix of DNA. The caduceus was not decorative. In Sumerian religious tradition, it was the personal emblem of Ningishzida, a god associated with the underworld, with healing, and specifically with what the texts call the good tree, a tree of life entwined with serpents and connected to the transmission of divine essence. The symbol also appears in ancient India, in Mesopotamian cylinder seals, and in Egyptian art. Cultures separated by oceans and centuries independently chose the same ascending double spiral to represent the force that animates living things. James Watson and Francis Crick confirmed the double helix structure of DNA in 1953. The Sumerians were drawing it more than 4,000 years before that confirmation. The structure is specific. It is not a loose spiral or a simple coil. It is 2 strands ascending in opposite directions around a shared axis, exactly as they do inside every cell of your body. That level of structural precision does not emerge from watching snakes climb a stick. Zecharia Sitchin, in his 1976 work, The 12th Planet, argued that Enki, the Anunnaki chief of genetic operations, used the caduceus not as a metaphor, but as a diagram. The academic establishment dismissed this without serious engagement. But dismissal is not refutation. The symbol exists. The structure matches. The timeline is impossible under any conventional explanation. What the Sumerians left on those temple walls was not art. It was documentation. They recorded something they had been shown in the only visual language they had, and they placed it at the center of their highest religious iconography. The question is not whether they drew the double helix. They did. The question is, who showed it to them? The bio-tablet. Around 2,100 years before the Common Era, in the Sumerian city of Nippur, a tradition of creation was already ancient. What survives in written form as the Atrahasis epic, a text committed to clay tablets around 1,700 years before the Common Era, does not read like a myth. It reads like a procedure. The gods, identified as the Anunnaki, are described as convening a council to address a labor crisis. Their solution is the creation of a worker species. What follows is a step-by-step account of biological fabrication that has no parallel in any other ancient mythology on Earth. Every mythological tradition has a creation story. Most involve breath, or clay, or divine declaration. The Atrahasis epic is different because it specifies. It names the presiding officer, the goddess Ninhursag, also called Ninmah, serving as chief medical authority. It describes the biological inputs, divine clay combined with the blood of a slain god, which Sitchin and subsequent researchers have interpreted as a genetic template merged with an existing biological substrate. It describes a protocol, and then it gives a number. 14. The text specifies 14 birth mothers, described in translations by scholar Stephanie Daly as womb goddesses, used in the initial creation batch. 14 is not a cosmologically significant number in Sumerian religion. It carries no symbolic weight in their astronomical or theological systems. It appears in this text exactly once, attached to a biological production run. In mythology, numbers serve symbolic purposes. 7, 12, 40. These are ritual numbers. 14 is a batch size. Jeremy Narby, a molecular biologist and anthropologist, argued in his 1998 work, The Cosmic Serpent, that ancient creation narratives encoded genuine biological knowledge transmitted through visionary states. He was not writing about the Anunnaki specifically. But his framework applies directly. The Atrahasis epic describes gene mixing, surrogate gestation, and iterative production. Those are not mythological concepts. They are laboratory concepts. Human chromosome 2 is the confirmed result of a fusion event between two ancestral primate chromosomes. That fusion has no gradual fossil record. It appears as a single edit in the human lineage. The Atrahasis epic describes a single deliberate intervention that produced a new species. One is a clay tablet. The other is your genome. They are describing the same event from different sides of the laboratory door. The ancestral code. 74,000 years ago, a supervolcano beneath what is now Lake Toba in Sumatra erupted with a force that dwarfed every volcanic event in human history. The ash cloud that followed may have triggered a volcanic winter lasting years. Anthropologist Stanley Ambrose, writing in 1998, proposed that this event reduced the entire human population of Earth to somewhere between 3,000 and 10,000 individuals. Geneticists, independently analyzing mitochondrial DNA, confirmed it. There was a bottleneck. We nearly ended. What emerged on the other side was not quite the same species that went in. Before the Toba event, anatomically modern humans had existed for approximately 300,000 years. They made tools. They used fire. They buried their dead. But they produced almost no symbolic culture. No art, no music. No evidence of abstract reasoning preserved in the archaeological record. For 200,000 years, the lights were on, but no one was writing anything down. Then, in what geologists call an instant, somewhere between 50,000 and 70,000 years ago, everything changed simultaneously. Language, symbolic art, ritual burial with grave goods, long-distance trade networks, musical instruments, and the first cave paintings appeared in the archaeological record within the same narrow window. Paleoanthropologist Richard Klein of Stanford called this the human revolution and proposed it was triggered by a neurological mutation. The specific mutation has never been identified. No gene, no chromosome, no sequence in the modern human genome has been conclusively named as the switch that turned behavioral modernity on. The timing of this cognitive explosion corresponds, within the margin of archaeological dating, to the Toba bottleneck. A population reduced to a few thousand individuals is a population at maximum genetic vulnerability. It is also a population that could be comprehensively modified and then replenished. If a genetic intervention were going to take place in the human lineage, a near-extinction event is not a complication. It is an opportunity. What emerged from the Toba bottleneck was a human being who could do something no prior human had done at scale. Think symbolically, speak in abstraction, plan across generations, and ask why. Science calls that a revolution. A revolution requires a cause. After 50 years of searching the genome for what caused it, the cause remains officially unknown. That is not a solved problem wearing the costume of one. That is an open wound in the evolutionary record, and the silence around it is its own kind of answer. The hybrid strand. In 2010, geneticist Svante Pääbo and his team at the Max Planck Institute for Evolutionary Anthropology published a paper in Science that rewrote the human family tree. By sequencing ancient DNA extracted from Neanderthal bone fragments, they confirmed what had previously been theoretical. Modern humans carry Neanderthal DNA. Not metaphorically, measurably. Between 1% and 4% of the genome of every non-African person alive today is Neanderthal in origin. The two species interbred. The evidence is in every cell of roughly 8 billion bodies walking the planet right now. Pääbo received the Nobel Prize in Physiology or Medicine in 2022 for this work. That finding alone was extraordinary. Then came the second one. In 2008, a single finger bone fragment recovered from the Denisova cave in Siberia was submitted for DNA analysis. The result identified a third hominin species, entirely unknown to science, that had lived alongside both Neanderthals and modern humans. The Denisovans. Their DNA survives today in Melanesian and Aboriginal Australian populations at concentrations of up to 6%, higher than the Neanderthal contribution to European genomes. In 2018, a single individual found in that same Siberian cave was identified as a first-generation hybrid, the daughter of a Neanderthal mother and a Denisovan father. Scientists named her Denny. 3 species confirmed in one cave, one of them a hybrid of the other 2. The human genome is not a clean evolutionary product. It is a composite document, assembled from at least 3 distinct lineages, stitched together in ways that formal evolutionary theory struggles to model with any elegance. Then comes the 4th lineage. Genomic analysis of sub-Saharan African populations has identified a statistically significant contribution from an archaic hominin that diverged from the human family tree approximately 500,000 years ago and interbred with modern humans as recently as 35,000 years ago. Researchers call it the ghost lineage. No fossil has ever been attributed to it. No tools. No cave art. No skeletal remains of any kind. It is present only as a signal inside living human DNA, a contribution from a species that archaeology has never physically found. We have excavated the bones of every hominin we shared this planet with, except one, and that one left the deepest genetic signature of all. Something was here, something that shaped the human genome in ways we are still mapping, and it vanished without leaving a single bone behind. That is not an evolutionary footnote. That is a deliberate absence. The Genetic Vault. In 2001, the Human Genome Project published its first complete draft map of human DNA. It was the most ambitious scientific undertaking in history, a decade-long international collaboration involving hundreds of researchers across 6 countries. The results were announced with appropriate ceremony. What was not announced with any ceremony, what appeared quietly in the body of the technical paper published in Nature that same year, was the identification of approximately 223 genes in the human genome, with no identifiable evolutionary precursor anywhere in the known tree of life. No equivalent in worms, no equivalent in flies, no equivalent in yeast. Genes that had, in the formal language of the consortium, no homologue in invertebrates. The standard explanation offered in that same paper was horizontal gene transfer, a process by which bacteria exchange genetic material with host organisms. Horizontal gene transfer is a documented and well understood phenomenon in microbial evolution. It is a legitimate mechanism. But the functions of these 223 genes are not microbial functions. They are not simple metabolic processes inherited from ancient bacterial ancestors. They include complex neurological signaling pathways, immune response modulation systems, and the synthesis of compounds unique to human biology. Bacteria do not build neurological networks. Bacteria do not design human cognitive architecture. Among the specific genes flagged in this category is the primary sequence governing the production of hyaluronic acid, the compound responsible for the unusual elasticity and self-repair capacity of human skin and connective tissue. Human skin is uniquely resilient among all primates. The gene driving that resilience has no clear evolutionary ancestor in any related species. It appeared, fully functional, in the human lineage, without a trail of intermediate forms leading back to anything simpler. Statistician Samuel Carlin of Stanford, one of the leading quantitative analysts in genomics, flagged this cluster of anomalous genes as statistically extraordinary. The academic conversation did not follow. Zecharia Sitchin cited the finding in his 2002 work, The Lost Book of Enki, as confirmation of the Anunnaki genetic intervention narrative. The scientific establishment dismissed Sitchin, but dismissing the interpreter does not make the data disappear. 223 genes with no evolutionary origin story are still in your genome. The Human Genome Project found them, labeled them, and the field moved on. That is not resolution. That is avoidance wearing the uniform of progress. Whatever put those genes there, it was not bacteria. And the fact that no serious institutional effort has been made to find another answer tells you something about how uncomfortable the real answer might be. The origin sample. In 1928, Peruvian archaeologist Julio Tello, excavating burial sites on the Paracas Peninsula of southern Peru, uncovered something that did not fit. The skulls of the Paracas culture, a civilization that flourished along the Peruvian coast from approximately 800 years before the Common Era to 100 years before it, were elongated. Not slightly, dramatically, and not only in shape, in volume. Standard artificial cranial deformation, the practice of binding an infant's skull to reshape it over time, can alter the geometry of a skull. It cannot increase its cranial capacity. Capacity is determined by brain size. The Paracas skulls had cranial volumes approximately 25% larger than a normal human skull. Tello documented the anomaly and moved on. For nearly 90 years, the academic consensus called it deformation and closed the file. In 2017, researcher Brian Forster submitted samples from 18 Paracas skulls to a laboratory in Texas for mitochondrial DNA analysis. The samples were handled with documented chain of custody protocols. The laboratory confirmed the samples were uncontaminated, and the results identified mitochondrial haplogroups, the genetic markers used to trace maternal lineage through human populations, that did not match any known Native American genetic signature. They did not match any Asian population. They did not match any European lineage. The mutation identified in the HVR1 region of the mitochondrial sequence was not present in any database of known human genetic variation. Physical anthropologist John Verano of Tulane University, the leading academic critic of alternative Paracas interpretations, has consistently argued that cranial differences can be explained by intentional deformation. He has not addressed the volumetric anomaly. You cannot bind a brain larger. That is not a detail. That is the entire argument. The DNA results were never published in a peer-reviewed journal. They were not refuted. They were not challenged in the literature. They were not followed up by any institutional research program. They simply did not become part of the official record. In science, an anomalous finding that cannot be refuted is supposed to generate investigation. What happened instead was silence, which is a different kind of answer. An unidentified mitochondrial haplogroup in a culture that built sophisticated textiles, practiced advanced cranial surgery, and left no clear genetic origin story in any standard anthropological model is not a coincidence. A DNA test found a lineage that does not exist in any known human population. No one published it. No one disproved it. It was filed under the only category that required no further explanation: inconvenient. The DNA relic. In 1982, cytogeneticist Jorge Yunes and his colleague Om Prakash published a paper in Science that identified something structurally impossible about the human genome. Every great ape on Earth, chimpanzees, gorillas, orangutans, bonobos, carries 48 chromosomes. Modern humans carry 46. 2 chromosomes are missing from the count that our closest genetic relatives share. The paper confirmed what the chromosome count already implied. At some point in the human lineage, 2 ancestral primate chromosomes fused together into a single chromosome. That chromosome is now called human chromosome 2, and the evidence of the fusion is still visible inside it. The confirmation came from the Human Genome Project. At the interior of chromosome 2, researchers found telomeric sequences, the repetitive DNA strings that appear exclusively at the ends of chromosomes, sitting in the middle of the chromosome instead. Telomeres do not appear in chromosome interiors unless 2 chromosomes were joined end to end. The stitching is still there. It is textbook cytogenetics. It is not disputed. Two chromosomes were fused into one, and the resulting human being had a genome incompatible with every other primate on the planet. Here is what the mainstream explanation does not fully account for. For this fusion to become heritable, it had to occur in a single individual, spread through enough individuals to form a viable breeding group, and those individuals had to successfully reproduce with each other, rather than with the far larger population of 48 chromosome hominins around them. Population geneticists have proposed mechanisms, including Robertsonian translocation and founder effects. Those mechanisms exist, but the probability of a spontaneous chromosomal fusion appearing in enough individuals simultaneously to establish a reproductively isolated breeding population remains, under any rigorous calculation, effectively negligible. Lloyd Pye, in his 1997 work, Everything You Know Is Wrong, was among the first to propose this fusion as evidence of deliberate genetic modification rather than random mutation. The academic establishment did not engage with the argument. It simply proceeded as though the probability problem did not exist. Your chromosome 2 is two chromosomes that were joined together. The join is visible. It happened once, in a single lineage, and produced a species that could no longer freely interbreed with its predecessors. Evolution accumulates change gradually. It does not perform single-generation chromosomal edits that redefine reproductive boundaries. What the fossil record calls the emergence of Homo sapiens, the genome calls a modification. The stitches are still in the DNA. Gradual processes do not leave stitches. The genome core. The Weld Blundell prism, housed in the Ashmolean Museum in Oxford, is a clay artifact from approximately 2100 years before the Common Era. Inscribed on its four faces is the Sumerian king list, a record of rulers stretching back to a time the text describes as before the flood. The reigns listed for those pre-flood kings are not measured in decades. They are measured in tens of thousands of years. 8 kings. Reigns ranging from 18,600 years to 43,200 years. After the flood, the list continues, but the lifespans collapse. Hundreds of years, then dozens, then the numbers we recognize today. Whatever those pre-flood figures represent, the scribes who recorded them did not consider them impossible. They considered them history. Modern genomics has confirmed that the human body carries the machinery for dramatically extended cellular lifespan. The TERT and TERK genes, along with the shelterin protein complex, regulate telomere length, the protective caps on chromosomes that shorten with each cell division. Telomere degradation is the primary driver of biological aging. What Elizabeth Blackburn, who received the Nobel Prize in Physiology in 2009 for this research, confirmed is that telomere shortening is not simply entropic wear. It is actively regulated. Something in the human body is managing the rate of aging. The system is not failing. It is being governed. David Sinclair of Harvard, in his 2019 work Lifespan, went further. He argued that aging is not biologically inevitable. It is a program, a set of instructions that, if altered, could be run differently. The maximum verified human lifespan, 122 years, achieved by Frenchwoman Jeanne Calment, has remained effectively unchanged throughout all of recorded history. Not because the cellular machinery cannot do better, because something is set to prevent it. The Sumerian king list does not record a gradual reduction in lifespan. It records a stepped, progressive decline across multiple post-flood generations, from tens of thousands of years to thousands to hundreds to the modern ceiling of roughly 100. That is not how mythology handles time. Mythology collapses. It exaggerates. It does not produce a controlled, multi-generational reduction curve that mirrors what a deliberate genetic modification rolling out across a population would actually look like. Your genome contains the hardware for a lifespan you are not being allowed to use. The switch that governs it was set a very long time ago. The Sumerian scribes recorded what life looked like before the switch was thrown. We are living in the after. And the distance between those two numbers, 43,000 years and 122, is not mythology. It is a settings change. The blood type anomaly. 15% of the human population carries a blood type that cannot be explained by primate evolution. The rhesus D antigen, present in the blood of all other primates and in 85% of modern humans, is simply absent in the remaining 15%. Rh-negative individuals do not carry the antigen. They did not inherit it from any primate ancestor science has identified, because no other primate lineage on Earth has produced an Rh-negative variant. The Rh-negative blood type is a genetic orphan. It exists without an evolutionary parent in any known species. The geographic distribution of Rh-negative blood is not random. Its highest global concentration is found among the Basque people of northern Spain and southern France, where prevalence reaches up to 35% of the population. The Basque are also genetically and culturally anomalous in a second independent way. Their language, Euskara, is a language isolate. It shares no root, no grammar structure, and no demonstrable ancestry with any other language on Earth. In a continent where every major language traces back to Proto-Indo-European roots going back 6,000 years, the Basques speak something that came from nowhere identifiable. Two profound mysteries, genetic and linguistic, concentrated in a single population. The reproductive biology of Rh-negative blood presents the most difficult problem. When an Rh-negative mother carries an Rh-positive fetus, her immune system identifies the fetal blood as foreign and mounts an attack. The condition, called hemolytic disease of the newborn, can be fatal to the child. Under standard evolutionary logic, a blood type that causes mothers to attack their own offspring should have been eliminated from the gene pool thousands of generations ago. Natural selection is ruthless about reproductive disadvantage. Yet Rh-negative blood not only survived, it maintains stable prevalence percentages in populations across multiple continents. A 2000 paper published in the European Journal of Human Genetics identified unusual neurological characteristics in Rh-negative populations at statistically significant rates. The research did not receive wide coverage. Coverage is not the same as validity. The human immune system is the most conservative biological system in the body. It is calibrated over millions of years to distinguish self from foreign. It does not make philosophical errors. It does not get confused by mythology. When an Rh-negative mother's immune system identifies her own child as foreign, it is not malfunctioning. It is reading the child's genetics accurately, and what it is reading in the blood of that child is something it was never taught to recognize as entirely its own. That is not an evolutionary quirk. That is a biological record of origin, the star sequence. In the 1930s, French anthropologist Marcel Griol began a decades-long investigation into the cosmology of the Dogon people of Mali. What he documented, confirmed in a formal publication co-authored with Germaine Dieterlen in 1950, was a body of astronomical knowledge that the Dogon could not have possessed. Their oral tradition describes Sirius, the brightest star in the night sky, not as a single star, but as a binary system. They named the companion star, described its orbital path as elliptical, specified its orbital period as 50 years, and identified it as the heaviest and most dense object in the heavens. Sirius B, the white dwarf companion to Sirius A, was not photographed by Western astronomers until 1970. Its orbital period is 50.09 years. The Dogon were accurate to within a fraction of 1%. The mainstream response, most notably from Carl Sagan, was contamination. The Dogon must have encountered Western astronomers before Griol arrived and absorbed the knowledge through contact. Anthropologist Walter Van Beek conducted follow-up fieldwork in 1991 and found that general Dogon community members showed limited awareness of the Sirius B knowledge. But Van Beek's own data confirmed what his conclusion tried to obscure. The astronomical knowledge was restricted to an initiated priestly class. Knowledge kept by a priesthood does not appear in general community interviews. That is the entire point of a priesthood. Robert Temple, in his 1976 work, The Sirius Mystery, calculated that the Dogon's astronomical data required an observational baseline of several centuries minimum, far predating any plausible Western contamination. And in the Dogon's own account of where this knowledge came from, there is no ambiguity. It was given to them by beings called the Nomo, described as amphibious entities who descended from the sky in a vessel accompanied by fire and sound. The Nomo are called the monitors of the universe in Dogon cosmology, and are specifically associated with what the tradition names the coil of life, a spiral symbol representing the generative force of existence. That symbol, preserved in Dogon religious artifacts centuries before James Watson was born, is visually identical to the double helix. Bring it all the way back to where this list began. A Sumerian stone carver drew the double helix in 4,500 years before the Common Era and called it the emblem of the god who created humanity. A West African priesthood preserved a spiral they called the coil of life and said it was given to them by beings from a star we could not photograph until 1970. Those beings gave the Dogon a number, 50 years. The number is correct to 2 decimal places. You cannot guess an orbital period for a star you cannot see. You can only know it if someone calculated it and gave it to you. The Dogon are specific about who that was. They came from Sirius. They descended in a vessel. They gave humanity the coil of life, which is the structure of DNA, and they left. What the Sumerians recorded as a diagram on a temple wall, what the Dogon preserved as sacred cosmology, what the Human Genome Project found without explanation, inside 223 genes, what your chromosome 2 shows in stitching that has never been accounted for. These are not separate mysteries. They are the same testimony, preserved in different languages, on different continents, across thousands of years, all pointing at the same event. Someone came here. They built something inside us, and then they left us the symbols to find our way back to the question. 10 items. 10 separate lines of evidence, drawn from molecular biology, archaeogenetics, ancient texts, physical anthropology, and oral tradition, each one anomalous on its own. Together, they form a pattern that conventional history has no clean framework for. The caduceus drawn before DNA was discovered, a creation text that reads like a lab protocol, a behavioral revolution with no identifiable genetic trigger, a ghost lineage that shaped the human genome and left no bones, 223 genes with no evolutionary origin, an unidentified mitochondrial haplogroup in skulls too large for binding to explain, a chromosomal fusion that should have been probabilistically impossible, longevity genes actively suppressed in a species that carries them, a blood type with no primate ancestor that the maternal immune system treats as foreign, and a West African priesthood holding the orbital period of an invisible star, given to them by beings who also gave them a symbol that looks exactly like the structure of life itself. The scientific establishment has an answer for each of these individually. It calls them coincidence, contamination, mythology, or convergence. But the accumulation is the argument. At some point, the number of coincidences required to maintain the conventional story becomes its own kind of impossibility. Go to the comments. Pick the one item from this list that you think is the hardest to explain away and make the case. Tell us why that specific piece of evidence is the one that holds. The floor is yours. If this changed how you see the record, subscribe. The archive goes much deeper than this, and you have only just started asking the right questions.